Hi, my name is Linda Goodman. I'm the Chief Technology Officer of Fauna Bio. We're a small six-time full-time employee company, and today I'm going to be talking about how really exciting biology in animals like lemurs, squirrels, and bears is inspiring new potential therapies to treat human disease. Many people in the audience are probably aware that there's been a dramatic drop in the cost of genetic sequencing over the past 15 years. And this has really opened the door to be able to identify new drug targets based off highly powered genomic studies. But the key problem with this has been that our vulnerability to common human diseases like heart disease, obesity, cancer, diabetes, really depends on hundreds, if not thousands, of mutations in our genes. And this has made it really difficult to isolate ideal drug targets. But there are a few people who have rare genetic superpowers that can protect them from common human diseases. For example, about 1 in 40 people of African ancestry has a mutation in the PCSK9 gene that leads to very low levels of LDL cholesterol. LDL is, of course, the bad type of cholesterol that you don't want. And this means that these people have an 80% lower risk of heart attack, which has led to successful inhibitors for this gene, PCSK9. And this means that now that we know there's this really impactful gene, we can all take advantage of this exciting biology with new therapeutics. But unfortunately, humans don't have genetic superpowers to cure every single disease. So at Fauna Bio, we are looking to animals to identify genes that might have a very large impact on human health. We are, of course, not the first people to come up with this idea. You may have seen in the popular press that animals like elephants don't get cancer. And there are many researchers combing over the elephant genome to try to identify genes that could be relevant for the next cancer therapy. You've also probably heard about the naked mole rat, which is a rather unattractive animal, but it defies the laws of aging. The mortality of a naked mole rat is in fact independent of their age. So researchers are also looking at the genome of this animal to try to identify a new aging therapy. Perhaps less well known is the 13-line ground squirrel, which is a hibernating animal that is able to protect itself from many different diseases that I'll get into. And the genomes of animals, particularly mammals, are highly relevant to humans. For example, we share about 92% of our genes with rodents. To give you a few examples of how these hibernating animals are relevant to human diseases, they're able to combat conditions like obesity and diabetes, heart attack, irritable bowel syndrome, Alzheimer's disease and stroke in the brain, and perhaps most intuitively, muscle and bone loss. You can imagine that these animals are lying motionless in hibernation for 6 to 9 months out of the year. But then when they wake up in the spring, they're able to immediately run from predators and have very minimal muscle and bone loss. The same could not be said of us if we were to lie on our couch for 6 to 9 months. We would not be able to walk after that. And so it's not just small animal hibernators. You've also probably heard of bears that are great hibernators. Really, every major mammalian clade has a hibernator, including primates. The fat-tailed dwarf lemur in Madagascar is a great hibernator. And these animals, in general, use the same genes to protect themselves from the conditions that they suffer during hibernation. And these genes are also present in humans. So this is a body temperature trace of the 13-line ground squirrel throughout the year. I think when many people consider hibernation, their impression is that these animals go into a deep sleep for the winter period. But in fact, for small animal hibernators like the 13-line ground squirrel, it's a very dynamic process. So they start out at around 37 degrees Celsius, where they are in the summer and the early fall. And their heart rate is about 300 beats per minute because they're a very small animal. And as they enter hibernation into a state called torpor, their temperature drops to 4 degrees Celsius and their heart rate drops all the way down to 3 to 5 beats per minute. And around 25 times throughout the winter, they have what are called IBAs, or interbout arousals, where over the course of 1 to 2 hours, their tissues get reperfused very suddenly with blood and oxygen, and their heart rate skyrockets again to 300 beats per minute, and all of the tissues get rewarmed to 37 degrees Celsius. And they do this about 25 times, and it's very akin to an ischemia reperfusion event that humans might get with a heart attack or stroke. So we like to say that hibernators experience 25 heart attacks per year, yet they have very little damage. For example, here are some slides of cardiac tissue from a rat, a squirrel during the summer, and a squirrel who is hibernating that have experienced damage. And we can see the white parts in the damaged tissue are leukocyte infiltration, so that's showing exactly how much damage there is. And the rat has the most damage, but the summer squirrel actually still does get quite a bit of damage. The most protected by far are these hibernating squirrels that are protected from these ischemia reperfusion events. So at Fauna Bio, we are investigating the genes that are turning on and off between the summer time point and the winter time point to better understand how humans might be able to protect our hearts from heart attack and stroke. These animals are also relevant for Alzheimer's disease. As they enter hibernation, their neuronal spines actually retract. You can see that in the top entrance picture, and they lose connectivity. They also develop hyperphosphorylated tau proteins, very similar to an Alzheimer's patient. Yet when they arouse, their neurons regrow, they regain the connectivity, and they can clear the tau protein. And so if we could understand the genes that are responsible for this, that are turning on and off at these time points, this might help us develop a therapy to reverse Alzheimer's disease. Hibernators also have very precise metabolic control. As we discussed, they about 25 times during the winter, they're revving up their metabolism and then tamping it down to go back into torpor. They also, as all good hibernators do, they bulk up and get nice and fat before hibernation. And they actually enter hibernation being insulin resistant. And then as they're leaving and they wake up in the spring, they're svelte and insulin sensitive again. So at Fauna Bio, to understand this hibernator disease protection, we start with hundreds of genomes and thousands of gene expression signatures. And so this is a snapshot of our data that's on AWS. Fauna Bio has the largest hibernation biobank in the world with 163 animals, 22 tissues sacrificed across 11 time points that are critical for hibernation. And we mine this data for transcriptomics and also epigenomic data. We also have the largest collection of mammal genomes in the world through our collaboration with the Broad Institute. And this is 142 species, which include 31 hibernators. And as you can imagine, these are very large databases, several terabytes in size each. And these would be quite challenging to analyze on our own personal computers. And so we are excited to be running all of our analyses and integrating them on AWS. Here's an example of the type of analyses that we do. So this is a phylogenetic tree. We can see the mammal ancestor at the bottom, and these are derived species that exist in the present day. And the branch length that we see between the species is equivalent to the number of mutations that have accumulated. And so on this tree, the hibernators are the bat, the ground squirrel, the tenrec, and the mammal ancestor. And what we like to do is identify genes that are highly constrained in the hibernators, but have been accumulating potentially damaging mutations in non-hibernators. So they might So they might look like that. So they look like that, sorry, where they get longer. So these have accumulated more mutations in the non-hibernating species. And this is what the results of that look like. So the hibernators are shown here in green, and they have a very high similarity to the sequence in the mammal ancestor, whereas humans and the non-hibernators have accumulated a lot more mutations. And in some cases, these genes are actually completely broken in humans and non-hibernators. And based on homology, we think that these genes may relate to diseases like Alzheimer's, diabetes, and bone loss. And here's an overall schematic of how our analysis works on AWS. We have our hibernator data sets with the RNA-seq and the ATAC-seq, which is epigenomic data, the hibernator evolutionary rate analysis that we just talked about, and we overlap these to identify the genes that come up more often than we'd expect by chance. We also like to include human genomic data, both common and rare variants, to look for networks that are enriched for human disease associations to make sure that our targets are going to translate. We also spend a lot of time curating external transcriptomic data sets from unprotected species because we like to make sure that the way that the genes are getting expressed is different in animals that are not protected as compared to our squirrels that are protected. And overlapping all of these data is how we identify our top targets. Fauna Bio has programs across 4 key tissues, and I'm going to start by talking about our program in the heart. And this is an actual screenshot of our AWS hosted platform for drug target identification. So ourselves, as well as our collaborators, can log on and select first the type of data that they'd like to work with, if it's comparative genomics, transcriptomics, epigenomics, or all 3. Then they can select the type of tissue that they're interested in designing a therapeutic for. And then, of course, the type of therapeutic. Is this supposed to save tissue from ischemia or reperfusion, or is it supposed to reverse Alzheimer's disease and have neurons regrow? And they can make all of these selections. And then the outputs look like this, where it'll select the top gene networks. So these are groups of genes that get upregulated and downregulated together at key time points. And we use the hubs of these networks as potential drug targets. And so in this specific network, these are genes that are downregulated during hypoxia in heart tissue, similar to if you had an ischemia reperfusion event, like a heart attack. And so this specific target is called Fauna 119G. And from there, the computational team passes it to our wet lab to test this target to see if it's functional. And so our wet lab team ran these experiments in cardiomyocytes where they deprived the cardiomyocytes of oxygen and examined if there was any effect compared to controls. And we see that knocking down this gene does indeed double human cardiomyocyte survival in vitro. We next wanted to see if we had a good understanding of the network. So we moved down to a gene that's one downstream, Fauna 120G, and this gene as well, a knockdown was able to greatly improve cell survival. And currently we're working to develop compounds to inhibit these genes to be a new potential cardiac therapy. Last, I'm going to talk about our very recent work in the liver with our partner Novo Nordisk. Liver tissue is highly relevant for satiety and obesity. So Novo Nordisk, our partner, is very interested in understanding the Biggest Loser syndrome. I'm sure many of you have seen or heard about the TV program The Biggest Loser, where contestants that start out being significantly overweight lose a large amount of weight through diet and exercise in a short period of time. The problem is that many of these individuals end up regaining the weight after the show is over and potentially even more weight than they started with. And it's not entirely their fault. There is unfortunately persistent metabolic adaptation 6 years after the Biggest Loser competition. Specifically, the resting metabolic rate had actually decreased by 610 kcals per day. And this makes it very difficult for individuals to maintain the weight loss. And so what Novo Nordisk and we would like to do is better understand how we can alter someone's metabolic rate to have it be more reflective of what it originally was. And as we discussed, hibernating animals are actually a great study system for this. 25 times throughout the winter, they're revving up and turning down their metabolism, and we're going to study these specific time points that they're adjusting that. Not only that, but they're a great model for appetite and satiety. As all good hibernators do, they eat a ton. In the fall, there's a specific time where they're actually eating 57% of a 24-hour day. But then 2 weeks prior to entering hibernation, they actually stop eating. And so they're satiated during that time period. And so we've started looking at some of this data, and this is very preliminary. But here's one of our early gene networks for looking at how we might go about increasing an individual's metabolic rate. And we see this hub Fauna 304G that's strongly downregulated during this time period where metabolic rate is going up. And we noticed in literature with humans that high levels of this gene are actually associated with obesity in people. So we're going to begin further investigating targets like this. And I'd like to thank the organizers of the conference for inviting me to speak today. And of course, I'd like to thank the rest of the Fauna Bio team for contributing to this research. Thank you very much.